Cutting-edge research laboratories are using multiplex immunofluorescence as a powerful tool to extract information from tissues. One data point at a time, spatial biology weaves a contextual map over biological tissues to track biomarkers, processes, and cell interactions. These maps provide a tremendous amount of information that is of fundamental importance to answer key scientific questions about a number of diseases. Extracting this information is complex, long, and challenging. But now, the way researchers like you face the complexity of spatial biology can radically change with an easy, fast, and accessible solution. With COMET, it is now possible to perform high-throughput, hyperplex immunofluorescence with the antibodies you already know and have characterized. So you can build on your know-how. Understanding biological processes is complex, but generating data doesn't have to be. COMET is the fastest high-throughput solution for spatial biology, all in one. COMET is fully automated, providing you with high-quality and reproducible data to accelerate your work. With the COMET end-to-end workflow, simply insert a sample and retrieve your images. With COMET, you can use any characterized clone from your library. You can move fast, develop your protocols within days, and get hyperplex results in a matter of hours. The unique technology is what makes all of this possible. COMET streamlines protocol design and optimization for you by organizing highly multiplexed assays into independent steps. Once each biomarker is optimized, they can be easily combined to generate ready-to-run protocols. The sample is inserted together with a microfluidic chip that integrates staining and imaging. Via a sequential immunofluorescence protocol, primary antibodies are dispensed, followed by fluorophore-labeled secondary antibodies. Then the slide is imaged, and the antibody complex is removed with a gentle elution step. The cycle starts again with new antibodies specific to different proteins, with outstanding preservation of the tissue morphology and epitope stability. And our revolution works. Accelerate your research, eliminate the complexity, and be among the first to join the COMET revolution. Lunaphore, accelerating spatial biology.